Hey, welcome back to the online Causal Inference seminar. Today, we're excited to have Maria Glymour from Boston University, who will talk about evidence triangulation in dementia research. After that, we will have a discussion by George Davey Smith from the University of Bristol. Questions today are handled by Georgia, so I'll quickly switch over to her.
Thanks, Dominic. As always, please submit your questions through the Q&A. Maria does not have a collaborator, but she has told us that she's willing to stop when questions come up and answer them. Please do not use the chat to ask your questions. That said, we can give the stage to Maria.
Thank you. It's a pleasure to be here. Thank you very much for the invitation. Let's see if I can pass the first challenge of any talk, which is sharing my screen successfully. All right. Can you guys see that? Great. All right. Thanks again. I'm Maria Glymour. I'm a chair of epidemiology at Boston University School of Public Health. And these slides were prepared with Serena Gutierrez, who is a PhD student at the University of California, San Francisco. And Serena, unfortunately, couldn't be here today, but I want to acknowledge her contributions to my thinking on this and this presentation. All right. I want to talk a little bit about evidence triangulation and in the context of dementia research. My assumption is that this audience is not necessarily super familiar with dementia research per se. And so I want to talk a little bit about some of the specific challenges in dementia research, which I think is like of the health domains, it's one of the more messy domains. And then I'm going to talk about a couple of examples that sort of fall broadly within evidence triangulation from my own team's work. One is sort of classic evidence triangulation, and the others are maybe a little bit less traditional, but I think you'll see the idea is using complementary sources of evidence to better understand a causal structure. And then very briefly, I'll touch on what I see as challenges. So to introduce, and again, feel free to ask questions if it's useful to clarify. There are lots, the talk is perfectly, like it's modular. I can skip parts if we have more discussion. So let me just talk about a little bit about dementia to give people a little context here. Dementia, the diagnostic criteria for dementia change over time, and at any given time, there are various different diagnostic criteria in use, but there are some common features of all diagnostic criteria for dementia. That there is cognitive function, cognitive performance below normal for the population, that cognitive performance of the individual has deteriorated from their prior performance, and that cognitive loss causes a functional loss or impairment, such as an activity of daily living limitation, like difficulties balancing your checkbook or cooking or whatever it is that you do in your life. So those three criteria are essential to dementia, and we can think about dementia more like disability than like a disease, i.e. it's a functional outcome that can be caused by many different pathophysiologic processes. There's Alzheimer's, of course, cerebrovascular disease, which we think of in its most acute form as stroke, and many other pathologies that occur in the brain that ultimately manifest in dementia. Alzheimer's disease dementia refers to the clinical syndrome that arises as a consequence of the Alzheimer's pathophysiologic process, which is specifically assumed to be due to amyloid deposition. I also want to emphasize cognitive impairment in most settings is detected and diagnosed through a combination of history taking from the patient and a knowledgeable informant, and an objective cognitive assessment, either a bedside mental status examination or ideally a comprehensive neuropsychological battery. So there's an objective measure of test performance. So with that background, it's important to understand that dementia really is a consequence of multiple superimposed processes. There's a process that is a developmental process that is, of course, very rapid in childhood, but also occurs throughout life, synaptogenesis, selective remodeling. There are disease processes. There are multiple disease processes, which often co-occur. We now think most dementia has multiple contributing underlying pathologies. And in fact, brains are quite active organs, and acute or chronic neurologic injury is followed by neurologic, cognitive, and behavioral compensation. So there is, in truth, reorganization. I think this little picture is basically before and after a stroke. So there are actually functional changes after the death of part of your brain. So with this background about dementia, there are a lot of features of dementia that truly, it's like they are put in there intentionally to defy efforts at causal inference to identify prevention opportunities. One is the disease develops slowly, insidiously over the course of probably decades. And in fact, the full course is a little bit unclear. There are, as I mentioned, multiple likely synergistic disease processes that contribute. It is very difficult to measure the onset of disease because it is rarely an acute and sudden onset. It is a slowly developing process. Subtle cognitive, behavioral, and physiologic changes emerge slowly. The diagnosis timing is, in fact, influenced by many other characteristics besides the actual functional status of the patient, including co-occurring illnesses, access to care, and educational attainment, for example. And as a result of this, biases in various studies will differ. For example, electronic health record-based studies where there's sort of passive data collection and detection of dementia diagnosis will have very different biases than studies where there is active outreach at designated time points to assess cognitive status. The other key issue about dementia is numerous confounders across the life course are deeply socially patterned. The confounders that I think are almost most systematically worrisome and omitted from most of our research studies are early life, quote unquote, IQ. And you can think about, you can have all kinds of critiques of IQ, but the basic concept that their cognitive skills that individuals have, and that influences a whole bunch of other behaviors. It also influences their dementia risk. And life course socioeconomic status is truly very strongly connected. And at some point, I took all the major risk factors that that are prioritized in most of the literature for dementia, and I looked back, and basically every one of them is very strongly related to childhood IQ or childhood cognitive status and childhood socioeconomic status. So in studies of dementia, because the average age of onset is in your 70s or 80s, in most studies where there has a lot of dementia occurring, there's not good prospective data on these important confounders. Okay, so that's why dementia, we have some causal inference challenges in this domain, this health domain. Now I want to move to the question of evidence triangulation. And I think this audience probably has a firm, formal, and intuitive grip on these concepts. So I will go quickly about what we mean by evidence triangulation. I think the most influential recent articulation of this was Debbie Lawler's paper, in which she describes evidence triangulation as strengthening causal inferences by integrating results from several different approaches, where each approach has different and assumed to be largely unrelated key sources of potential bias. More recently, Jeremy Lebrecht and Sonia Swanson wrote, I think, a nice paper in Current Epi Reports, trying to be a little bit more formal by what we mean by evidence triangulation. I actually think it's a little more, I have a more flexible view of what should count within the evidence triangulation umbrella and what's needed in the field, but I think their framework is actually very helpful. The idea is that you have multiple estimators or two estimators. If you apply both estimators to the same data and research question, we would expect them to agree when the causal assumptions are satisfied for both methods. And you have to compare estimators that, given substantive knowledge, are expected to agree only when they are both unbiased. So I think that's a very helpful framework, even though even if we think we can learn things from data evidence triangulation that doesn't fully satisfy that. Is evidence triangulation new? I would say, of course not. The motivating premise of triangulation is not so different from Bradford Hill's consistency criterion. This is a quote from Bradford Hill. The consistency of the observed association, i.e., has it been repeatedly observed by different persons in different places, circumstances, and times? The lesson here is that broadly the same answer has been reached in quite a wide variety of situations and techniques. In other words, we can justifiably infer that the association is not due to some constant error or fallacy that permeates every inquiry. And we have to indeed be on our guards against that. This is like, I feel like this should be an intro epi for every intro epi class. The same results for precisely the same form of inquiry will not invariably greatly strengthen the original evidence. I would myself put a good deal of weight upon similar results reached in quite different ways. That is the core of evidence triangulation, of course. And in general, this concept is familiar in many scientific processes. Why is there sort of sudden attention to evidence triangulation? I think, in fact, I've thought about this, and George and I talked about this, and I'm interested to hear his thoughts. I think, in fact, in observational health research, we've really been dominated by a particular format of study and for years. And this is an example of the evidence triangulation, the pyramid of evidence that many of us encountered early in our research methods training, which I think implicitly or explicitly has really framed how we think about evidence. I think modern causal inference frameworks help us specify assumptions that we actually need to draw causal inferences, and in ways that historically, the previous decades of risk factor epi often did not do. And once you've articulated those assumptions, it's easier to appropriately compare estimates from different studies and more systematically pursue evidence triangulation. I think the better data, more data opportunities offer more opportunities for triangulation and help us rule out consistencies due to chance. And the evidence pyramid and dominance of a particular study design has led to oversimplification of causal inferences from the field as a whole, i.e. assumption that we climb the ladder to the best possible research design, a randomized control trial, and then draw inferences from that design using meta-analyses. And increasingly, the inadequacies of this framework are clear, and that it doesn't really acknowledge how the specifics of how a particular cohort study or case control study, the key assumptions might be very specific, and something higher in that hierarchy doesn't necessarily address the key assumption. I don't think we can really talk about evidence triangulation without discussing meta-analysis, because if you think about evidence triangulation as basically a way to synthesize evidence, meta-analyses are probably the predominant tool that epidemiology and health research has for evidence integration. But meta-analyses are intended to combine estimates from similar studies. And by combining estimates, we make chance and increasingly less plausible explanation for our finding, but it doesn't really help us address the assumptions of the underlying studies necessarily. Let's see, there's a question. Will the recording?
Don't worry about that.
Thank you. Okay. I'll keep the Q&A open so I can actually see if there's a question that I can answer, because I can't answer that question. A lot of evidence triangulation has really focused on alternative identification approaches. And I think that causal identification is not the only way to triangulate, the only key type of evidence triangulation, but I think it is really core and has driven a lot of the conversation. So when we think about all of the tools we have for identifying causal effects, you can kind of think of them as falling into three big buckets: blocking the backdoor criterion, blocking all backdoor paths, which conditioning on common prior causes. I refer to these as covariate control methods in general with epi. I think it kind of matches the language that epidemiologists are most familiar with. A second approach is to use an instrumental variable. And the third approach is fulfilling the front door criterion. As of now, I think this doesn't come up a lot. I have high hopes, and maybe someday it will feature more widely in our work. But I think as of now, we're really looking at studies that use covariate control methods or studies that use instrumental variables. And observational health research has really been dominated by backdoor criterion-based approaches, while economists tend to lean much more on IVs or similar kinds of approaches. Let's see. All right. Dementia research in particular has been really dominated by backdoor or covariate control approaches. It's very hard to do randomized controlled trials for prevention of dementia for all the reasons I mentioned. A recent major review published in The Lancet, led by Dr. Livingston, sort of went through many different risk factors from early life. This picture here has made population attributable fractions famous in dementia research. From early life, like education, going on through mid to late life, like diabetes and hearing loss. Although there's a sort of hope about these risk factors being very well established. In fact, if you drill down, for the most part, they are almost all reliant on studies that are covariate control-based studies. There are a handful of quasi-experimental studies, which for example, for education and hearing aids, but for the most part, this research is based on covariate control. And in some cases, for example, estimating the effects of alcohol use, there's no quasi-experimental evidence yet on the effects on dementia. And the backdoor results, the covariate control results are selectively ignored because people are uncomfortable with the conclusions, right? So if you actually look at most observational studies of alcohol and dementia, it looks like moderate alcohol use is substantially healthier than no alcohol use. But people are quite uncomfortable reporting that because they're concerned about the health implications of promoting even moderate alcohol use. And so there's really a kind of brushing of results under the rug. So I think what's happened is we have so little confidence in many of our methods that we're actually not willing to take our results to the bank unless we already agreed with them before we did the science, and we think that the results are either harmless or would be beneficial for other reasons. This is, of course, not a great situation for science if we want to actually discover ways to prevent dementia. And I think an evidence triangulation approach can help us a lot with potentially ruling in or out these various risk factors. I want to go briefly into some specific categories of approach that people do. The question is, do they draw a DAG and try to control and check the backdoor criterion, or is it just a metaphor for saying they try to control for confounders? They try to control for confounders. There are certainly some studies that use DAGs and articulate the DAGs, but in general, no. In general, people are just controlling for confounders. And the field of epi is still very heterogeneous to the extent people are worried about controlling for potential mediators or factors influenced by the exposure, and people who are not. So it's very routine in your paper reviews to get reviewers saying you need to control for all these things that are obviously downstream of your exposure, and then you have to have fight back to say why you didn't do that. Yeah, so if you really push people, the way that they think that they're doing causal identification is they're fulfilling the backdoor criterion, but they certainly are not using that language, and often they're not using DAGs, but sometimes they're. Hopefully that's clarifying. Okay, so if we just think about the classic backdoor criterion-based approach where we are going to measure an exposure, we're going to measure the outcome, and we're going to try to control for confounders, and we think about that as sort of the conventional approach, what are some evidence triangulation approaches that can move beyond that or strengthen inferences from that type of study? As I mentioned, there's meta-analysis where you combine multiple studies with similar designs. I think a variation on meta-analysis that is actually much more useful is meta-regression, where you use features of the contributing studies to evaluate sources of heterogeneity in the meta-analyzed estimate. The meta-analysis helps rule out chance, but it can't rule out biases. The idea, in fact, you're taught with meta-analyses to try to find very similar studies to meta-analyze. And so clearly they're going to have similar biases. Meta-regression, I think, really can help us understand which types of assumptions are key to driving patterns of exposure. Another approach is cross-context comparisons, where we really think the potential confounders are different. So we have a setting or a population where the most plausible confounders are just quite different than they would be in our first study. This is, I think, is actually a very promising approach that, as we have more and more data from very heterogeneous settings, can be quite powerful. Then there are instrumental variable-based approaches, and I put them all together as randomized control trials, because I think for epidemiologists, it's really important to emphasize that Mendelian randomization or genetic IV and policy-based IVs, they have the same implicit, we hope for the same causal structure as an RCT. It's just that we're not sure if we've actually fulfilled the assumptions that we have. And exactly the assumptions for IV really matched the things that we worry about in a randomized control trial. So randomized controlled trials in which there's intentional randomization, but then instrumental variables analysis using genetic variants as the instruments, so so-called Mendelian randomization. This, I think, Mendelian randomization has been incredibly influential and has really changed how people thought about study designs. But of course, long before there were genetic IV, there were IV analyses using other policy variations. And when I say IV analyses here, I don't just mean 2-stage least squares estimates of the IV parameter. I think that, in fact, you can use the idea that you have an instrument to assess whether there's any causal effect, whether that causal effect is the sign of that causal effect, and so on. These are, I think, policy variations are very promising, but of course, actually, there are lots and lots of very clever instruments that are not based on policies. And then the last approach that I think is very important and promising is to really focus on actually demonstrating bias. And this is where, in some ways, the Lebrock-Swanson framework, it kind of doesn't capture this, but I actually think it's a really important part of evidence triangulation, is trying to say what how do we think we could show the bias or quantify the bias? So that includes looking for negative control exposures, negative control outcomes. So these are situations where you use a different variable with a well-established null association with the exposure, with the outcome, but a similar confounding structure. And then if you observe that this is in fact a null association, there is in fact no association between your negative control and that suggests that the confounding is also not large. Evaluation of reverse causation. I think this is a particularly important application of Mendelian randomization. I'll talk about an application in a couple slides. And quantitative bias analysis, which in our group we found super helpful in that a lot of potential biases are clearly potential biases, but when you actually try to write down the parameters of a causal system, of a data generating model that could produce a pattern as large as you see, it's really quite hard to reproduce it with plausible effect sizes. And we in fact do have a lot of constraints or frameworks for sort of saying these are plausible effect sizes, and we can rule out certain types of bias fully accounting for patterns. All right, with that background, I'm going to talk about a few actual examples coming out of my group, the first of which is about education and dementia. And I think the education and dementia example is the closest to like sort of classic evidence triangulation. And then I'll talk about three other examples that are a little bit less traditional. Okay, so example one, the research question is, does educational attainment prevent dementia or delay incidence of dementia? There is very strong, longstanding evidence that people with more education have lower incidence of dementia. And this is one of actually several meta-analyses that come up with very strong associations showing here the meta-analyzed risk ratio is around 0.4. So, but nearly all of those studies share the assumption of no confounding by prior cognition or parental socioeconomic status. And they're all essentially covariate control-based studies, and they all have that same assumption. However, there were huge changes in mandatory schooling in the United States across the 20th century. And those huge changes led to very large increases in educational attainment. And Claudia Golden, before she was I think so famous around women's work, actually did really wonderful work on educational attainment. And this is showing radical increases in secondary school enrollment and graduation across the United States. This is white graduates, white 18-year-olds, I think, across the course of the first half of the 20th century. You can see these huge increases. Much of that was enacted via changes in mandated schooling, so the age when children could drop out of school. This is a plot just for the state of South Carolina, where you can see for birth cohorts born in 1925, they were only required to complete 6 years of school, but children born in 1926 would have had to go to 8 years of school. And then in 1934, they moved it up to 9 years of school. So you can see these really create quasi-experimental opportunities. This is Tu Nguyen, who's now on faculty at the University of Maryland, but was at the time a postdoc. I think she was a postdoc, but she might have been a pre-doc when she did this work. Where she used both, in the data from the health and retirement study, she estimated the association between educational attainment and absolute risk of dementia. And then she used a genetic instrumental variable, and then she used mandatory schooling laws. And she used all of these to estimate the effect of a year of education on dementia risk. And essentially within these samples, she found in all cases, there was a pretty strong positive, there was a pretty strong benefit. The estimate, and I think this is an important discussion point, the estimate from the policy-based IV was huge and probably much larger than plausible as a causal effect of educational attainment alone. And I'm happy to return to that in discussion if people are interested. I think it's a super important phenomenon. That basic result has been replicated in many, many settings across the world and in diverse data sets. And additionally, actually, since we did that work, covariate controlled methods have improved in their capacity. We've got data sets that now have early life IQ, for example. So some of the most important confounders have also come into data sets, so we can control for it. And that result is quite consistent. There are still very important questions about the potential that education doesn't really help you truly functionally, but it just makes it much harder to diagnose dementia. If you remember, all of the dementia diagnoses are based on performance on these little tests. If there's one thing education does for us, it helps us take tests better. Whether that really pays off in terms of functional stuff is, I think, a little bit less clear. But that's an important. And I guess another issue is sort of the consistency of patterns across settings where social advantages conveyed by education vary. So, for example, in the United States, there are huge increases in educational attainment of Black Americans. Black men and Black women experience very different social benefits from that education in the 60s to 70s due to racial discrimination, in particular against Black men. So understanding to what extent education actually has a benefit when you can't sort of get the social benefits as much. So that was my first example. The second example, and this is a little different use of this approach, but I think you can see how it falls into the evidence triangulation framework. This is an example where we were really interested in the question of at what age do dementia-related diseases begin to influence physiology? So as I mentioned, we have this is a long, slowly progressing disease. We need to understand when the disease really begins, because we need to know that to understand prevention. If you want to prevent something, you need to start before the disease starts, right? So we need to understand that for primary prevention. It will help us understand when interventions aren't working, why. There is a potential for confounding in associations of time-varying risk factors, such as BMI, body mass index, hypertension. We need to understand when reverse causation starts in order to understand what would be a sufficient set to identify causal effects of these time-varying exposures. And also very much in the topic of controversy is the role of amyloid as the leading indicator of disease. And that has been important. This is work that has been led and completed by a bunch of wonderful team members, and now Willa Brenowitz, who's at Kaiser Portland, Scott Zimmerman, Peter Buteau, and Min Choi, who are all research analysts on my team, have all contributed importantly to this work. We've done a few examples of this. We started with body mass index because there was a sort of funny puzzle, but now we've done a few other examples, but I'm going to talk mostly about body mass index because it's has the prettiest picture. Midlife obesity is a risk factor associated with increased risk of dementia. So we know that people who are obese in midlife have higher risk of dementia in later life. Alzheimer's dementia, however, is associated with weight loss at around the time of diagnosis. This is a very well-established association among patients. So when does dementia-related weight loss begin? You can see that this is quite challenging because you have a causal effect that's increasing risk and an effect of the disease that is decreasing the exposure value. So this is work originally led by Willa, where her idea is to use a genetic instrument for Alzheimer's disease to disentangle this. And you can think if you're looking at people who are of an age before the dementia process has begun to influence weight loss, any Alzheimer's disease genetic risk should be unrelated to weight loss. They're just totally not connected. And once Alzheimer's disease begins to influence, once there is preclinical dementia, so the Alzheimer's disease genetic risk starts to trigger dementia-related processes, the Alzheimer's genetic risk should be associated with weight loss. That's just saying these predictions. So the idea here is to say, at what age does the Alzheimer's disease genetic risk begin to be associated with weight? We used the UK Biobank to originally investigate this. The big advantage of UK Biobank is it's a very large sample. So about 400,000 people. So we have a genetic risk score for Alzheimer's. The genetic risk score for Alzheimer's is actually pretty strong. Mostly there's one allele that's very strong and then a bunch of other alleles that are small, but together strongly relate to Alzheimer's disease. Body mass index measured at study enrollment. And then what we did here is we estimated originally, as we've done this, we've like kind of improved our approach, but originally we estimated a linear regression of BMI on age and age squared, because that's actually the functional form of how BMI changes as people get older. They first gain weight and they kind of peak and begin to lose weight. That's just if you plot the age pattern of body mass index. And then we specified an interaction with the genetic risk score. We subsequently, I think, improved on that by basically allowing the line for the AD genetic risk score to begin to interact with age across different ages, and we chose the age at which it was the best fitting to show that there's a change in the age trajectory of that exposure. Okay, so for this initial one with BMI, we're looking at BMI and whether the association with age is modified by the genetic risk score. And then I'm just going to show you curves for people at the 10th and 90th percentile of Alzheimer's disease risk. And what you can see here, so this is the red line are people with low Alzheimer's disease genetic risk, and the black line are people with high Alzheimer's disease genetic risk. And what you can see is before around age 50, there's really no association between Alzheimer's disease genetic risk and BMI. Like it's just, as you'd expect, the genetics of Alzheimer's have nothing to do with BMI. Until basically late 40s, early 50s, these start to diverge. And you start to see that weight loss is faster in people. And I should say, this is all cross-sectional data, right? So this is the big caveat here is it's cross-sectional data. So, but effectively the age trajectory of BMI becomes much more shallow for people with high Alzheimer's disease genetic risk. This was in UK Biobank. We're just recently fielding it. Like the biggest concern here, of course, is the selection process in UK Biobank. We're just recently confirming this in all of us. We're just writing up those results now, and the preliminary results show us a very similar pattern where you see people with the Alzheimer's disease genetic risk becomes relevant in kind of late 40s, early 50s, and you start to see people having lower BMI if they have high AD genetic risk after midlife. And in fact, we then went and looked at the cognitive assessments in UK Biobank. They have a bunch of weird cognitive assessments in UK Biobank that I don't know how they ended up with these, and there's samples for all of them differ. So we looked across all these cognitive assessments and tried to find the ones that there was an age-related, there's an age-related change in them that's very standard, and we tried to find if there were ones that the Alzheimer's disease genetic risk accelerated age-related change, and at what age did that begin? So that's essentially the same model as what you saw with BMI, but we've done it with a bunch of different cognitive outcomes. And essentially there are 12 different cognitive outcomes. And what this is showing is age from 40 to 70, what the best fitting age of divergence was, suggesting that basically some of them it was too young to detect in UK Biobank, like we never saw a period where there wasn't an association. But many of them around late 40s, early to mid 50s, you started to see the AD-GRS is predicting accelerated declines. So that was, So the sort of conclusion here, these findings suggest just Alzheimer's disease related changes in body mass index, cognition. We've also looked at brain MRI measures beginning to emerge in late 40s, early 50s. Of course, there are lots of caveats here. This is a strange sampling process. This is completely cross-sectional data. The changes could be attributable to pleiotropic effects of genetic variants. I don't think that's the best explanation but it's certainly a plausible explanation. But the implications here, if these is true, is that Alzheimer's research beginning at age 65 is at least 20 years too late for primary prevention. And risk factor associations for measures in middle age or later are potentially confounded by emerging disease. And this latter point is a huge issue for, use it or lose it research. Like there's a gazillion studies of playing brain games and playing Sudoku and how that affects dementia risk, clearly potentially biased by these processes.
Maria, before we move on to the next example, there is a question that I think is related to this age example. Are there any special causal approaches in studying dementia for longitudinal data apart from uses of IVs?
I would say the number one challenge outside, if you're just thinking about covariate control methods, I think the number one way to improve dementia research is not to use dementia as the outcome, which a perspective that doesn't make me super popular always in dementia research, but I think that the most powerful way to look at, like the fundamental feature of dementia is that it causes cognitive deterioration and loss. And I think looking at within-person change in cognition over time is a much more powerful framework and useful framework than looking at incidence of dementia. Incidence of dementia is profoundly and inescapably associated with where you started cognitively. And so you just can't get around that very strong relationship between early life cognitive development and cognitive resources and dementia risk. And so I think work focusing on dementia as an outcome very often basically delivers all of the predictors or things that predict cognition by age 25. And that's important. It's useful, I think, but it's not about Alzheimer's disease. That's about a different health outcome, which is cognitive development. So I think the number one thing for causal inference related to dementia is to focus on within person changes in a continuous measure of cognitive function. And yeah, let's see. So the third example I want to talk about is an example focused on this very funny observation that people who have survived cancer have lower incidence rates of dementia. And by incident, I'm using incidence rates. I'm being very specific there. It's cases per unit time. It's not that if you die, you're less likely to develop. If you have cancer, which is a potentially life-threatening disease, you're less likely to develop dementia before you die. It's that for any given unit of time that you're alive, you have a lower incidence rate of dementia than people who've had no cancer. The one obvious explanation, which I absolutely immediately spat out as soon as I read this association, is it's selection bias due to death. That's ridiculous. That obvious explanation is not sufficient to explain several lines of evidence, including that this association is observed in non-fatal cancers. So cancers that really wouldn't kill you still have this pattern. And I want to emphasize this is very interesting, whether cancer itself is a cause or more plausibly that there's a shared biological process that increases cancer risk, i.e. uncontrolled cell growth, but reduces dementia risk, which is essentially inappropriate neuronal death. So we set out to try to understand this using a few different approaches. And this is work led by multiple colleagues. Monica Espina-Romero, who's now a neuropathologist at the University of Wisconsin, Lindsay Kobayashi, who's on faculty at the University of Michigan, Jingshuan Wang, who's a wonderful PhD student at UCSF, Elizabeth Rose Maeda, who's a completely brilliant dementia epidemiologist at UCLA, and Eleanor Hayes Larson, Elizabeth Rose's postdoc, who's about to begin a faculty position at USC. We wrote a bunch of papers related to that. And I'm going to go through kind of how these papers variously tackled this question. So the first thing, which Monica and Lindsay, Monica led and Lindsay was senior author, really tried to use a meta-regression approach. So she pulled together evidence from multiple different study designs. And the key, I think, a really important contrast, there were a few different contrasts, but a key contrast was studies that she thought were very vulnerable to confounder bias because they didn't have good confounder lists, studies that she thought were very vulnerable to diagnostic bias, studies that would be very vulnerable to competing risks, survival bias, and so on. And then estimated the hazard ratio across each group of studies, excluding the others. And in fact, the log hazard ratios, excluding studies weak in each area, range from negative 0.09, so about a 10% protection, to negative 0.32. And the key upward bias, so the bias making cancer look less protective, was from studies when cancer status might influence AD diagnosis. So when in fact, if you had cancer, you were more likely to get diagnosed with dementia, regardless of your underlying cognitive status. The key downward bias, making studies look more beneficial, was from survival bias. So for example, when prevalent and incident cancers were combined. We also tried a negative control approach using spousal rate of memory decline as it related to the other spouse's cancer history, and found almost no association with the negative controls. So that was a little bit encouraging, using the spouse's trajectory as a negative control. Elizabeth Rose oversaw quite a nice set of bias quantification simulations with simulation models using incidence rates from observed data to sort of inform model parameters. And competing risk and selective survival in sort of the classic sense did not create a substantial bias that could have plausibly explained the association. But an important issue was there is often a delay between when somebody has dementia and the time when they get diagnosed, right? So they have dementia, they have impairment, but there's a period of time when they're living with dementia but haven't been diagnosed. Death from cancer during that period of time could easily cause a very large bias. So that's an important potential problem. And then we also looked at tried to just directly quantify diagnostic bias. And this was work led by Jingshuan Wang. And essentially what we find, if you engage with the healthcare system for any reason, if an older adult engages with the healthcare system for any reason, you are way more likely to be diagnosed with dementia. Like, as far as I can tell, these old folks are wandering into their doctors and the doctors are like, you know what, it hadn't occurred to me before, but now I'm going to assess you for dementia and diagnose you. So there is a substantial upward bias immediately after you engage with the healthcare system for almost any reason. This is actually, I'm just going to go through this. I'm not going to spend time on this because this is Monica's bias-specific meta-analysis showing sort of how much the estimate varied across these studies. So this was just like a really fun set of studies. And I guess the take home is, for the question, do factors that cause cancer reduce the incidence rate of dementia? I actually still think that that's quite plausible that could be true. However, it's extreme. The magnitude of bias from studies in electronic health records is so large. I think we could almost never detect it in those studies, and we need quite a different study design. All right, my last applied example is work done by Sarah Ackley. And I will say Sarah last week took a faculty job at Brown. I'm so excited for her. She's been a postdoc with my team, but I'm excited for Brown too. So Sarah has done a set of papers on evaluating medications with shared biological mechanisms using instrumental variables. And this is probably the furthest afield from evidence triangulation and its classic framework, but I think it's a really nice application of these tools. We've applied this in the context of amyloid targeting medications for Alzheimer's. These have been in the news. You've probably heard about some of these. For a long time, the primary target of medications for Alzheimer's treatment has been amyloid reduction. And there's a set of monoclonal antibody medications that target amyloid and remove it from the brain. And the premise of Sarah's work here was to pool information to derive an estimate of the effect of amyloid removal. All the drug trials, of course, are using intent to treat estimates for the effect of being randomized to receive the drug. And for the most part, people randomized do receive the drug, but the drugs have very heterogeneous effects on amyloid reduction. Some of them were very ineffective and some of them are quite effective. And so what we'd like to know is, suppose you actually take out amyloid, what does that do to cognition? Or dementia risk. So studies were reporting grouped data by randomization arm. There are often multiple different doses in a trial. So there's lots of different values if you think about randomization as an instrument, there are lots of different values there. And a cognitive measure in Sarah's original work, the most common cognitive measure was the MMSE. And for a subset of individuals, they would also report data on the amyloid change achieved within each arm. So they can talk about the biomarker change. This in some ways could be a straightforward IV analysis. It's a little bit challenging because the data are grouped and there's uneven sample sizes. So there's some limitations to what you can do because the companies are not sharing their individual level data with us. If anybody has them, please try to do a real analysis with the individual data. So here's the DAG here. We've got people randomized to various doses of drug from 0 to different intensities. That influences amyloid reduction. We want to know about the effect of amyloid reduction on cognitive outcomes. This was her original, on the left is Sarah's original work, which was published. The applied analysis was published in BMJ with an accompanying, just brief tutorial on the concept. And essentially the IV does this beautiful thing where it says, you expect the cognitive change to be proportional to the change in amyloid. And across drugs that had very small changes in amyloid, you expect very small changes in cognition, and right. So this essentially found a null result with some confidence interval. About a year after Sarah's original paper was published, there were a series of trials, some of which were null and some of which were resoundingly some of which found benefits published. So Sarah just then did a sort of Bayesian-informed meta-analysis updated with these new trial results. And she did this in this really nice way where she said, suppose you started with a prior that was from my meta-analysis. Suppose you started with a prior that you said the meta-analysis was nonsense, there was all publication bias, there's nothing. And you had the same width as our meta-analysis did, the same uncertainty bounds. Suppose you were a pessimist and you really thought these drugs are terrible and the companies are lying to us. This was your prior, was that there was a harmful prior. Or you were a true believer and you said, oh, you thought that they were actually beneficial and you started with a prior that was like, I can't remember exactly how we chose 0.055. It was like the most optimistic, plausible at that point. Here's the posterior estimate you'd get after you integrated these 4 new trials. And essentially, it doesn't matter very much where you started. The 4 new trials offered so much more data that we have a pretty good estimate of what amyloid reduction would do for cognition within the time frame of the trials, which is a small benefit. That, of course, can't rule out the possibility that there are, it can't totally rule out the possibility that there are practice effects, for example. So the take home here, evaluating mechanisms with a shared biological mechanism, there are caveats regarding biological heterogeneity of amyloid species targeted by the drug. I will say at this point, there's no statistical evidence to support such heterogeneity in that the instrumental variable estimates are consistent across the drugs. There's a lot of uncertainty, so we can't rule it out, but there's no, people keep saying, oh, this drug worked because it's targeting a different thing or it's doing something. It's like, that's totally unclear. This drug had a larger effect on amyloid, so it's unclear that it's actually important what species it targets. There are other challenges due to aggregated data that I mentioned. In general, this seems like a very straightforward complement to standalone trial data, and it's valuable to temper interpretations of chance results from single trials. There's a question here. Would you suggest as a follow-up or diagnostic steps when counterintuitive causal association? Oh, I will come back to that. That's like my last slide. So are there studies here that deal with heterogeneity and treatment effects and how? Bayesianism is one way of dealing with this. Yeah, I think there's no reason not to look at subgroups. And I'm not showing you this, Sarah did specific medications that were tried in multiple trials and then different categories of medication. So you could subgroup this however you wanted and look at the estimates. I think, yeah, there are other problems with this related to accessing the data. I would think that's the biggest problem with this method is accessing the data. Most trials of amyloid targeting medications, the data were never published. All right, I have like one more slide on challenges. So, I think it's actually quite challenging to really do evidence triangulation in its perfect sense. And one big challenge is how do you actually combine estimates across studies? And there, I think we need a lot more focus on being very systematic about how we combine things to say, are these the same estimands? And I think there's a tendency to say these are not the same estimand, full stop, but actually like, okay, can you convert them to the same estimand? Is it reasonable to think that these different estimands would come up with very similar estimates? That is a substantive question, of course, but I think that it's not enough to just say, oh, they're different estimands. We don't expect them to be the same. Is there the same exposure definition? This actually turns out to be a huge issue where, as far as I can tell, there's almost no constraints on variations in how we define exposures. And then trying to integrate evidence across different exposure definitions can be challenging. Same outcome definition. This is a disaster in dementia research that we have like you'll see all these meta-analyses, like there's a meta-analysis of cognitive change and then separately meta-analyses of dementia incidence. Really, everybody cares about dementia and why how that causes cognitive change. Like we think cognitive change is mostly occurring because of dementia, but they're being totally separated because there's not an easy way to integrate the estimates. Are these the same populations or do we expect real heterogeneity? And then once we get to that, ideally, we will be able to combine results or use quantitative bias analysis or other bias assessments to correct before we quantify. All right, I will stop there so we get to hear a little bit from George and maybe hear some questions. Bottom line, I think interest in evidence triangulation is growing, but we really need a sea change in the level of enthusiasm for finding out we were wrong about our research. Computational tools and data resources are also growing, and honest statements of why we're uncertain are really essential. I want to thank my amazing team, National Institute on Aging, for funding us. Melodem, if people are interested in methods and longitudinal research, it's a really great setting to get feedback. All right, I'll stop there. Thank you. George.
Yeah.
Thank you. And thank you for dealing with the Q&A. So yes, George, you may start now. Thank you.
Yeah. So yeah, that was a fantastic talk. I mean, I disagree with not very little of it, which is Maria and I have talked quite a lot about it, attempting to write something together. So we've had a lot of discussion. So I'm not going to have a lot of disagreements. I'm going to I would have some discussion on some of the examples, which I think were strong and interesting. But I'm going to just talk about some more general issues. So Maria said, said maybe I'd say something about why I think evidence triangulation has taken off. Which, I mean, if you just purely do a sort of Google Scholar look at it, and epidemiology, then it's absolutely taken off relatively recently. I think there's two issues there. One is that in ecological epidemiology, we're interested in evidence triangulation. The concept of triangulation was really introduced in social sciences in different contexts, whereas causality in many of the examples wasn't the issue. So there, for example, there's a lot of work on triangulation, on data triangulation, which would be sort of saying, how do you just get estimates of the sort of prevalence of something? And there's some very interesting examples in HIV, for example, where you're not actually asking a causal question, you're just trying to say, how do we actually get good estimates of HIV in different situations where there's different biases, different selection issues, different populations studied, and we're trying to get an estimate of the prevalence in the overall population, for example. So it's been used in lots of different fields, but I think it's entered ecological epidemiology as an ecological concept. I mean, I think there's Maria's talking about dementia. I think there's space for these other forms of triangulation which aren't trying to answer sort of necessarily causal questions. For example, in terms of the diagnosis of the different subtypes of dementia, how you can make some sort of classifications based. Some probabilistic classifications based on looking at how the genetic score relates to the dementia, for the genetic score for Alzheimer's versus the genetic score for cardiovascular disease, for example, relates to the cases, and use that and use that for for some form of differentiation of groups. So I think epidemiologists would do well to revisit forms of triangulation which aren't triangulation around causal questions. I have one view on why it's taken off. Is because of the popularity of modern epidemiology as a textbook, which in the first edition was just totally denigrated Bradford Hill's criteria, said that specificity was worse than useless, and as people were influenced by that book, they're not going to. They have something else to say. They don't want to reference Bradford Hill, or seem to be naive to folk who carry on with this antiquated notion. And so triangulation is a way of actually using some of the same sort of thought patterns, but without actually referencing Bradford Hill. I agree very much with Maria that the essence of it is in much of what Bradford Hill wrote, and not just in his paper on his viewpoints. He never referred to them as criteria. In fact, he explicitly said these are not criteria. They were sort of informal ways of trying to get better evidence about causation. Secondly, I mean a key issue in triangulation of evidence, so triangulation of causal questions, is the orthogonality of bias. So are the biases in different study types truly unrelated to each other? Now, you can have a sort of informal way of addressing that, which is just saying that logically, it's people, if you know about the sorts of studies that are used and what they're being used for, you can have a quite strong notion, and you can do some sorts of quantitative looks at whether the biases triangulate. So, for example, take LDL. Let's take a very obvious question where no one sensible continues to argue about, which is whether ApoB containing lipoproteins, LDL cholesterol, cause coronary heart disease. And two study designs there, I think powerful study designs there would be randomized controlled trials of different agents which reduce the ApoB containing lipoproteins through different mechanisms. So you have randomized trials, and we have a huge number of those with many different agents that substantially reduce LDL cholesterol, and Mendelian randomization, where you have substantial amount of genetic data for genetic variants which influence LDL cholesterol levels. I mean, it's an interest that now there's no reason why various things could bias the trials, differential lots to follow up, that people who react in a particular way to the drug and stop taking it, they're the ones who actually would have benefited least or most, or even been harmed by it if they'd carried on taking the drug. You have all those possibilities, but of course, those possibilities would not influence the results of a genetic study, a Mendelian randomization study, estimating the effect of LDL cholesterol on coronary heart disease. I mean, it raises the very interesting issue about the estimand, because of course your estimand in a randomized trial is 5 years, say, of difference that is generated by the treatment on cholesterol levels. So you get what is the effect of 5 years of having a different cholesterol level in middle age on your coronary heart disease risk. The Mendelian randomization estimates are of a difference from birth. You can show that the genetic variants related to LDL cholesterol relate to proportional differences in LDL cholesterol, pretty much the same in umbilical cord blood from the newborn and in 7-year-olds in Alzheimer and etc. The proportional effects are roughly the same. So you're looking at a lifetime, a postnatal lifetime difference, and there might be an antenatal effect, a postnatal lifetime difference in LDL cholesterol. Now, this one is a very nice example, I think, because you can scale the effect differences. And the bottom line is that basically 5 years of treatment gets you about 40% of the effect that you see in a Mendelian randomization study. And fascinatingly, that is the same across all the different drug types. The longer the trials carry on for, the greater the effect becomes, etc. But you can scale it to 5 years. So that's a situation where you can actually scale the two estimates, and then, in principle, combine scaled estimates, which are scaled to each other. Another example of orthogonality of bias, as Maria mentioned, the raising of the school leaving age, is in UK Biobank. You can look at that very issue because the school leaving age rate was raised in 1972 in the UK. It led to about 6 months more years of education, because a lot of kids who would have left at 15 didn't leave at 15, had to stay on by law. Now, there's no reason how that estimate, regression displacement estimate, would in any way influence what you get from a Mendelian randomization study of the effect of education on a whole host of outcomes. And Neil Davies led a paper that was just out in the International Journal of Epidemiology, comparing those two approaches at estimation. Now, both of them actually give you larger estimates than the observational estimate. And I'm a bit interested in Maria's view on this, because to me, the extent, you say, well, IV is correct for measurement error, which I agree with, but I think I've done a little bit of playing, we haven't published any of this, but a little bit of playing around with how much measurement error there can be. and the degree to which the estimates are larger than the observational estimates, to me seems to be more than you could get through measurement error correction. But those are two examples where I think that that you have a good reason to think that the biases will not relate to each other. And secondly, you can look at it. So in UK Biobank, for example, you can look at the genetic scores predicting educational attainment in those before and after, in those who finished their education before or after the 1972 change. And there's no difference in the genetic score until you get to even in the very oldest, where you get selection, because higher educational attainment means you die less. There's not enough. There was not enough by the time of recruitment to generate anything. A major issue is with incorporating biological data, which you can't convert into estimands. I think that an issue with triangulation is of course publication bias, because there's so many different methods you can use that you can use a whole set of negative controls, and one could select the ones which look best. So I'm an advocate. I know there's a lot of debate about this, but I am an advocate of pre-registration of triangulation studies. And I'll stop.
Thank you so much. And Maria, we are, as you heard, late on time, but would you like to respond in just 1 minute?
I agree, and I think the issue about education is whether you consider something measurement error in the endogenous variable or an exclusion restriction violation is really a substantive thing. So if you think about education as like, oh, we didn't measure years of educational attainment correctly, then measurement error is not going to explain this. If you think about education as the set of contextual experiences and enhancement that you get from being in a classroom, I think then that's a different thing. And I think that's probably what's happening with the IVs for education, is that what you get from being in school is not nearly adequately captured by years of educational attainment. But I think it's a really important problem, and I don't know. So thanks so much, George. That was great. It was really good to hear it.
Well, thank you both. Thank you, Maria, for your talk, and thank you, George, for the discussion. I apologize again for having to cut you off. I just want to make sure we stay on time as well, as much as possible. All right, so I will wrap up the seminar today, just reminding everybody that we have online Causal Inference seminar next week. We have 2 student presentations with Xinwei Shen from ETH and Giulio Grossi from the University of Florence next week. I hope to see you there. If your questions weren't answered, they will be forwarded to the speaker and the discussant. So thank you so much for being here.